History of our planet with microbes and evolution, the world that Darwin never saw. Available at estore.asm.org. Explore the fundamental role.
Welcome to ASM Live, the internet talk show live from the 2012 General Meeting of the American Society for Microbiology in San Francisco, California. I'm your host, Stanley Malloy. I'm the chair of the American Society for Microbiology Communications Committee and the dean of the College of Sciences at San Diego State University. Feel free to ask questions throughout our conversation today. You can use the chat function on the video viewer or send us questions by Twitter through hashtag ASM2012. Love to have you join in on the conversation with us today. In this session, we have with us Katie Kirsch from the University of Houston and Jonathan Eisen from the University of California at Davis. Katie, both of our speakers at this session are involved in thinking about microbes in places. And so Katie is a very unusual situation because she's here as an expert who just graduated as an undergraduate. So has achieved some really interesting things and got a lot of airplay by looking at microbes in hotel rooms. Could you tell us something about that, Katie?
Absolutely. Well, currently in the lodging industry, there isn't standardization with the cleaning. So the practices vary between brands and even properties within the brand. And so the cleaning methodology right now isn't based off of actual data. It's just what they think is best, is trial and error, essentially. And so what we wanted to do is create a very preliminary data set to evaluate the surfaces within a hotel room to identify the areas that are most likely to be contaminated so that for training and development of procedures, you can address those issues first, the high-risk areas, so that if there are any issues, they would be eradicated with the standard operating procedures.
And what were the high-risk areas? How is this a problem for us? Should we be afraid of hotel rooms?
Well, the lodging industry as a whole is doing a fantastic job. This project was made to better it. We didn't test for infectious disease, so if there were any diseases within the hotel room, not that we think that there was, we wouldn't have known based on our testing. We tested for aerobic plate counts, which are just general bacteria, pathogenic and non-pathogenic, and then also for coliform bacteria, which can be an indicator of fecal bacteria, which isn't really something that you want to think of in reference to a hotel room, but it's interesting to see. And so for the aerobic plate counts, the areas that were kind of concerning were the sponge and the mop, and what we did is test that on the made cards. We tested the mop, the sponge, and the glove. And the glove is disposable. Generally, they just use that for one room, and the counts weren't that high, but consistently the mop and the sponge were the two highest areas that we sampled throughout the rooms. And that presents a very high risk for cross-contamination because if you clean the toilet with the sponge and then go to the counter where you put your toothbrush, that bacteria can be transferred.
Yum.
Yeah. And then also areas like the toilet basin, the bathroom floor, then also the light switches and the TV remote keypad, so really the high-touch areas, which is sort of what we were expecting, but it was good to identify. And then also something interesting to note was that there was a high standard deviation among the areas. So if the TV remote in one room, for instance, might not have had a high count, but then in the other rooms might have been just astronomical. And that's an area of concern because it shows that the lack of standardization is affecting the levels within the room. The TV remote keypad in one room isn't getting cleaned as well as in another room, so that's just another argument for the project.
So is this one room that you looked at in detail, or how was your sampling?
Well, it was actually a collaborative effort between the University of Houston, Purdue University, and the University of South Carolina. And what we did is three researchers evaluated the rooms. So in South Carolina, Indiana, and Texas, we sampled one hotel in each state 3 hotel rooms within the hotel. So the sample size was 9, which is a very limited sample size. So it's important to note that this is very preliminary. Ideally, the sample size would be much larger. And then we sampled 19 areas in the hotel, each hotel room, and then put the data together to come with the mean and the standard deviation.
So you're staying in a hotel here in San Francisco. Are you collecting samples?
I wish. I wish. We don't have the equipment with us, though, so no.
So what's the outcome of this? Are you going to then make some recommendations that you think will change the process and improve the hotel industry?
Well, what we did for the project is we applied the principles of HACCP, which is a system developed by NASA, and it's widely used in the food industry. And what it does is it identifies and eliminates the risks for biological, physical, and chemical hazards that could potentially be there. And our argument is that that would be an ideal system to apply to hotel rooms. And so right now we haven't finished all of the system. We would need a little bit more communication with the industry to see how to better adapt it for the industry, but that would be our recommendation, is to apply the principles to your hotel room, and then hopefully eventually the lodging industry will get some sort of standardization.
So HACCP, just for our audience, is HAACP, right?
HACCP.
HACCP.
Hazard
Thanks.
Analysis and Critical Control Points.
Thanks. So one other interesting thing is, I mentioned that you were an undergraduate, you've just graduated, and your training was not in microbiology.
No, it wasn't. I actually took a class in food safety and sanitation at the Conrad Hilton College of Hotel and Restaurant Management. And I was really interested in what was going on, and we actually have a hidden gem, the food science lab there, which you wouldn't think. But this was just a project that was going on there, and I was fortunate enough to be able to join in on it.
That's very nice. And now so you've just graduated and you're going on. Are you going to continue to work in this area?
Yes, I am actually going to be attending Texas A&M in the fall for food science and technology for my master's, and I'm really excited because then I'll have a much stronger base of the microbiology. Right now I feel like I just got a taste of it, and I can't wait to start getting into the science classes. But then also the degree in hotel and restaurant management, having that side will help too, I think.
That's great. Wonderful. Now, Jonathan, you have spent a lot of time lately thinking about the microbiology of the built environment. What the heck is a built environment?
Yeah, so you could also ask, what is a microbe? So what I'm interested in recently is structures that are created by people. So anything that is built, a building, a car, a plane, a boat, a cave, if they modify it in some way, largely because they are sort of places we spend a lot of time, as well as places that are sort of plopped down into an environment and now are, depending on how they were made, starting out relatively microbe-free or microbe-low, and then they become colonized. So what I'm really interested in, and there's sort of a new field growing up right now, is treating the built environment as ecosystems. So the building that we're in now, we may not see a lot of non-human life around here. I think those plants are fake, but there's not a lot of non-human life in here, but there's lots of other organisms, including a lot of microbes. And the people interested in human health for a long time have done studies such as the one you've heard about with hotel rooms, with what microbes are present in buildings using a variety of methods, but it hasn't been focusing on the system as an ecosystem. It's been largely focused on where are the nasties. How can we prevent the nasty organisms from being there, and how can we kill them? And that's clearly an important component, but what I'm really interested in is the basic science of what regulates where microbes are in a building or a car or a plane, what regulates how they get around, and in particular, how does that affect the microbes that are in and on us, both the good and the bad and the indifferent ones. So sort of taking approaches that have been largely built up in ocean microbiology or studies of the human gut or soil microbiology, sort of microbial ecology, and applying those methods and approaches to studying the microbes that live in the places where we spend most of our lives.
So you mentioned the nasties. I mean, Katie talked about coliforms.
Yeah.
People very often talk about mold and mildew in environments. But if you take this analogy and think about the ecology of organisms, for example, our studies on the human microbiome, as you said, there's a lot of good bacteria and a few bad ones. What about in an environment like this room? Is it true also that in these places there are beneficial bacteria that make the room healthier? I mean, it's a very different perspective than most of us have.
Yeah, I mean, I probably would say, like with the human ecosystem, most of what we know is that there are a lot of things there, and most of them don't seem to make us particularly sick. Whether or not you could call them good or indifferent is still sort of up in the air with a lot of systems. But if you went to the savannah in Africa and you didn't want to get eaten, you probably would want to know where the lions and the cheetahs and maybe the hyenas or something else are out there. So obviously it's a concern to know where the things that make us sick are. But if you want to understand how the lions got there and what affects the numbers of lions in a system, you need to know where their food is. You need to know where the water is in the ecosystem. You need to know where the shelter is. You need to know where the other organisms are. And the same is true for microbial ecology. So it's not that individual organisms in this building are necessarily good or bad. It's that they all interact with each other. And only a small fraction of them make us really sick. Maybe a bunch of them, affect us slightly. And maybe some of them you could consider probiotics for a building or something that is beneficial. But really what we're at right now is we've focused so much on the nasty things. We don't know what the ecology is like in a lot of these environments, and we don't know how much to incorporate information about the interactions among microbes and the interactions between microbes and building surfaces in our studies of the microbes that affect us.
So what you have described and what Katie described, it's very interesting that one of the key differences is that Katie used a very traditional approach to culture the microbes that are there. And when culturing, you see this small subset, especially the ones that you're looking for. And you're describing a broader approach that's going to allow you to see everything that's there, or nearly everything.
Well, yeah, part of the reason people are interested in doing this now in buildings is that people have developed culture-independent approaches that include DNA sequencing and field microscopy and, proteomics and nanosims and all these amazing methods that people are talking about at this meeting that you could call culture-independent methods. But most of those methods have been applied to other ecosystems. And, for 30 years in the oceans and for 15 years in the human ecosystem and for, 5 years in the acid mine drainages or, other places, and for 2 years really in culture-independent studies of the microbial ecology of buildings are pretty new. And in ocean microbiology, the interesting stuff comes from linking the culture-based approaches where we can learn a lot about specific microbes to the culture-independent studies where we can learn a very little bit about a lot of microbes. And so it's bringing those approaches together that's probably going to be most beneficial, just as it has been in other ecosystems. So what my drive is to try and introduce building scientists, hotel management people, hospital scientists, people who build cars, people who look at planes, to introduce them, those building scientists, to the culture-independent approaches so that they can add that as a layer into their studies and become ecologists of their ecosystem. They already study hotels or restaurants or buildings or planes. And what I want them to become are ecologists rather than pathogen, focused microbiologists.
So I want to emphasize that we welcome questions from our audience and our online audience as well. We have a question right now.
Yes, we have a question from the video chat room. Iris asks, which method was used for identification of the bacteria? I know you talked about culture. I'm wondering, could you go more in detail on that?
We just did regular aerobic plate counts and then coliform bacteria counts using Petri film. We did environmental swabs that were put into peptone buffer solution, and then they were sent on ice to the University of Houston from the other states, and then we did the testing there with the Petri film.
So Katie, most people who have come through science classes sometime or other have used Petri dishes, and so they're familiar with that, but Petri film isn't something everyone knows about. What is that?
Petri film is fantastic. It's essentially just dehydrated Petri plates, and so they come and you just open the flap, they're pre-made, and they're just really quick and easy. I sound like I'm doing a plug for them right now. But it's pretty much the same thing, and then the coliform Petri film, it tests for just that. So it's selective media.
So it inhibits growth of non-coliforms. Yeah. So that's great.
Thank you.
Another question from the audience?
This is for Jonathan. Do you have any plans to look at the so-called sick building syndrome that we used to hear more about than I think we do today, but those instances seem to have been fairly well documented in the literature, but I don't know about if causes were really identified. Does that figure into your work?
Yeah, I mean, so for my specific work, where I'm more interested in the broad ecology of the built environment ecosystem, there are a large number of people who have started to apply culture-independent methods to a variety of known health problems in buildings, such as hospital-acquired infections and sick building syndrome. So there are labs out there that are using these methods right now to try and see if something about a building that is tagged by the people in the building, by their phenotypes or by epidemiology as being potentially a detrimental environment, is there something different about the microbes that are found throughout that environment? Very analogous to what people are doing with things like Crohn's disease or obesity in the human ecosystem, where they're looking for correlations. So that doesn't mean that there's a causative relationship. If you have buildings that are labeled as sick buildings, many of them, for example, are wetter than other buildings, and moisture is going to affect microbial ecology almost certainly. It may be independent, in essence, of the sickness that comes in buildings. We don't really know. So they may just be a correlated feature. They may be an actual causative relationship. What I'm involved in is trying largely to bring people together. So we have a project called MicrobeNet that's funded by the Sloan Foundation, and we're having a session this afternoon that she's speaking in on microbiology of the built environment. And the Alfred P. Sloan Foundation has a whole new program in this area. They've funded about 20 grants to people to do some studies like you are suggesting. Most of it is the basic science level still, because we really know nothing about the ecosystems. And it's very hard to go into an ecosystem and try and figure out what's gone wrong when there's sickness if you don't know the baseline. So if you're familiar with the Human Microbiome Project, that's been the reason why people have been trying to survey the baseline of the microbes in people, so that then, when people want to study what makes us sick or healthy, we can compare to the baseline. And that's what I view my role as now for buildings. I want to encourage people to figure out what the background is. What are the sort of general features of the ecosystem, so that then we can more easily determine when something goes wrong.
So Jonathan, it would be very interesting if you had some insight into some hotel rooms, so we could make this direct connection between the two of you. Have people looked at the hotel rooms with non-culture methods yet?
Yeah, there are people looking at, I mean, not just hotel rooms, but lots of living type environments and doing surveys similar to what you were suggesting with trying to pre-identify places that might have a lot of contact with hands or might be moist or near toilets or, and there's a couple of recent studies that came out, from Rob Knight and Jordan Peccia and a few other labs where people were looking at living residences. And there have been, I've heard of people doing hotel-associated studies. I haven't seen any published off the top of my head, but there are a lot of people that are trying to bring these new approaches. I don't know if this is the case for you, but I know that some people have had trouble convincing the people who own buildings to allow those darn microbiologists into their buildings in order to do some studies. We tried to start a project on some new construction where the builders were not dying to have us in the buildings. And I know that some people have had the same problem with hospital surveys and with restaurants. And so you have to find either a management school that is got their own buildings that they already work with, that they already study in some way that might be easier for you to get access to, or you need to find sort of a visionary builder or architect or agency that is willing to partner the microbiologists with the building scientists and the building owners in order to try and figure out what's going on. Because just imagine you go in there and with your little swabs, and I say I'm going to survey all the microbes in, some hotel room, and we find, an excess of chlamydia or an excess of something that the owners would probably not want you to be announcing into public. It's very touchy how you negotiate those types of interactions.
Mm-hmm. So Katie, did you have a problem with that? How did that work for you?
Well, the hoteliers that we were working with were eager to find out the results so that they could improve their training methods. It wasn't, the study was never something of us going in and pointing fingers or saying anything like that. And they really they're doing a great job, but they really do want to improve. And they recognize that this is probably the best way to identify the areas that need improvement. So they were eager to work with us, and they weren't necessarily associated with the schools. They were just hoteliers that were concerned about guest safety and wanted to do their best to promote it.
Do you think there's a difference between 5-star hotels and 4-star hotels and so on?
Not at all. I think it really depends on the management and the emphasis that they're putting on cleanliness, because in a 5-star hotel, when you have 500 rooms, sometimes you still have the same number of staff. And if the staff only has 15 minutes to clean a room, at a 5-star hotel where the room's bigger, versus maybe a 30-room hotel where they have 30 minutes to clean that room, you can't say just because it has more stars that it's going to be more clean necessarily.
I think it's important to reiterate something you said earlier today, and that is that detecting coliforms does not mean that someone in that room would get sick. Right? Absolutely. Mm-hmm. So I think that the potential is there, but it's not a direct cause and effect. And I think that ties into what you've been saying as well, Jonathan, that is that if you know everything that's out there, you're going to see some things that may look benign and some things that potentially could be less.
It's an interesting coupling of sort of culturing and culture-independent methods, because the culture-independent methods, the way they're done, a lot of times people first try and characterize what kinds of organisms are in a particular place using a variety of methods that are culture-independent. And then literally the first thing everybody wants to do in these environments is say, is that one going to make me sick? And all we have is a name or a genus. And within most genera of microbes, there are some that make you sick and some that don't. And even within individual strains, they make some people sick and other people are unaffected. So it's very hard from both the culture-based methods and the culture-independent methods to necessarily determine which ones are going to make you sick or which ones are going to have a detrimental effect on people. And in the long run, what we probably need to do is to start to couple these types of studies with epidemiology, where we actually know what the phenotypic effect is on people in those environments. And then what you're looking for is something that is associated with that effect, as opposed to going in and just surveying the microbes, and if I found something called, Mycobacterium tuberculosis versus Escherichia coli, can I say that those are going to make you sick? I think that in the long run, we need to actually couple these studies to health-associated phenotypes, if that's what you're interested in. I'm going to confess, I'm not interested in that. I'm interested in the dynamics of the microbes with each other. And that's still hard to study, let alone how they affect the people in the environment.
Well, I think that that's an important point because it influences the fear people might have of an environment.
Yeah.
And in fact, the studies that were just released over the last week on the microbiome, the human microbiome for healthy people, suggest that we're all carrying around some pathogens with us. Mm-hmm. And by definition, healthy people aren't sick.
Yeah.
And so I think that that's a very important point that people need to think about, mm-hmm. While at the same time we can improve the cleanliness of places and make the policies more acceptable. Mm-hmm. So in terms of thinking about the built environment, there are these changes that are associated with humans providing a unique microenvironment for organisms. But it's also true that we change the outside world as well. So for example, I know an area that you're interested in, Jonathan, is what's the difference between native species and between species that have been influenced by human agriculture? Could you say a little something about that too?
Yeah, I mean, so humans have obviously massive impacts on this planet. And some of those are, you could say, short-term, like you cut down a forest and you put a house there, and how that's going to affect the organisms in that environment and the temperature and the water flow and the microbes is what I'm particularly interested in. And, in terms of the planet itself, one of the things that we've changed on the planet is the lives of other organisms on purpose, right? So the domestication of cows and dogs and cats and plants, when we have shaped the lives of those organisms by breeding or genetic engineering now or whatever it is, much of the work that is focused on those organisms has been on the genes inside those organisms that are shaped by breeding. Like, what happens when tomatoes are made into, a certain color. And there hasn't been a lot of focus on what has happened to the cloud of microbes that lives in and on those plants and animals. And again, just like in the last couple of years, people have started to look at the cloud of microbes in and on people and how that has changed with diet and antibiotic exposure and movement across the planet, we probably should, and I certainly am, look at the cloud of microbes that lives in and on our domesticated food or partners or other organisms that are in our environment. First of all, the pets, those clearly affect us and our lives, both in terms of pathogens and natural microbes. So dog and cat microbiome projects, we announced a new kind of a joke, but now kind of serious thing called the Kitty Microbiome Project. Last week, Jennifer Gardy came up with this as a sort of way to attract.
At least it's not the Hello Kitty Microbiome Project.
I'm going to register that in a few minutes. But we live with these animals in our lives, and we have been studying the human microbiome, but we really should be starting to study the cloud of microbes that are in and on those animals. And then if you want to increase the yield, if that's your goal of, crop plants or animals, and for example, in cows, you're pumping them full of antibiotics, for example, there are people that are starting to say, what is that doing to the microbial cloud that is in and on those animals. Is that why, for example, there's an increase in incidence of E. coli O157H7? And so my lab and many others are interested in, again, taking these same culture-independent methods and starting to use them to characterize the domesticated plants and animals on the planet. And there are lots of people doing that. I mean, there are hundreds of talks at this meeting on people looking at every type of plant and animal on the planet, from ants to anteaters to, tropical trees to Venus flytraps to everything. We're going to now study the cloud of microbes that are around everything.
We have a question from the audience.
Yeah, actually, we have another question from the internet chat. And this goes once again back to hotel rooms. And that question is there any available number for specific bacteria levels for it to be safe in a hotel, like they have bacteria levels, safety levels for food?
Well, that goes back to our application of the HACCP principle. There's not a lot of academic literature on hotel rooms. And so we've developed a limit, but there's not really a way to verify it yet because our data set is so small and because there isn't a lot of other data to compare it to. So that it's still being worked on. But what we came up with, it's based on a study by Dr. Dancer, what she did for hospitals, and what she proposed was for coliform bacteria for less than 1 CFU colony forming unit per centimeter squared. And my proposal is that we double that to 2 colony forming units per centimeter squared, because I don't believe that for a hotel room it needs to be as strict. There will still be immunocompromised individuals staying there, but I just don't see the reason for it being that stringent. So we haven't verified that. We would need future studies to verify that, but that's part of the HACCP plan. So it's in the works with the project.
What about conference rooms?
Conference rooms.
Sorry. We're going to spend a lot of time in them.
Right. But that's an amazing thing, because you're talking about what is essentially one bacterium in a little square that's about this big, right? And that's a pretty low density when you're talking about it. Now, these are coliforms, but still, it's, in terms of human health, that's a very low density. You would have to be licking the floor over a wide area before you'd be likely to get very sick.
Yes.
So, another question from the audience.
This isn't really a question, it's a hooray for doing your work, because I travel internationally and I have been in some rather interesting hotels. My thoughts on hotel microbiology, building microbiology, and airline microbiology are really frightening for the general public. When I was last in, I don't know if it was Indonesia or Europe, it doesn't matter, I was on a long flight over the pond, and they played Contagion, the DVD.
Oh, no.
I wrote a letter to the CEO of United. I said, you're crazy to play that on a long flight. Well, what came out of it is I interviewed some of the flight attendants and some of the people, because I was really buzzed on it. It's fascinating how much they don't want to frighten the public. The flight attendants are told not to wear gloves because it might freak out the public. And somebody actively coughing isn't counseled. Could you either cough into your elbow or whatever? So my suggestion with your research, and both of yours, is to be proactive with the general public and say, excuse me, we do have flora and fauna in hotels and airports. Be proactive. Wash your hands frequently with soap and water. If you're traveling, carry one of those little antibiotic bottles with you, but I never use it unless I'm actually without soap and water. But to assume that a hotel, I love that, the 5 stars, no. It happens everywhere, and for the general public to get it, I would love to end my comment with a quote, if I could remember who was a famous parasitologist. It might have been Dr. Beaver who wrote This Wormy World. He started out with, man is both the diner and the dinner at the common feast. that's where we're at in the public world right now. So thanks for your research.
Thank you.
Thank you very much. So we're nearing the end of our program here. We just had a little plug for the movie Contagion. And I might point out that a scientific advisor for Contagion is going to be one of the people we interview in one of the later ASM Lives. So stick with ASM Live and you'll see that later on in the day. We will, after we finish this program, have another ASM Live coming up at 12 noon. So please join us again for that next ASM Live. And I'd like to thank Katie Kirsch from the University of Houston, soon to be from Texas A&M, for joining us today.
Thank you.
And Jonathan Eisen from the University of California at Davis. Thank you both very much.
Thank you.